Hello, I'm Shinwon Park, and today I'll be presenting on the shifting role of thalamocortical connectivity in the emergence of large-scale functional brain organization across lifespan development. Looking at these numerous atlases, have you ever wondered how do the distinct areas in the cortex acquire their function during development? This is one of the most fundamental questions in the field of developmental neuroscience. This has also been the subject of decades of seminal research on cortical arealization. Researchers have long debated whether regional diversity develops due to pre-patterned genetic influences or in response to input signals from the thalamus. Nowadays, it is widely accepted that both factors play an important role in cortical arealization. However, we must note that these principles that underlie cortical arealization typically refer to what is happening during the prenatal period. But since our brain continues to develop beyond birth up until young adulthood, we were interested in how these dynamics change across development, particularly in the role of thalamocortical connectivity in shaping large-scale functional brain organization. The thalamus, which was traditionally known for passively relaying sensory information to the cortex, is now increasingly recognized for its active role in cognition. Meanwhile, the organization of low-level sensory processing and higher-order cognitive functions is embedded within the principal gradient of macroscale cortical organization, which represents a hierarchy ranging from unimodal to transmodal brain regions. So, given the involvement of the thalamus in both external and internal processes, it is natural to question its role in shaping brain functional architecture during development. I will give you an overview of the methods and strategies that we used in order to answer this research question. First, lifespan neuroimaging, where we utilize the dimensional reduction techniques to obtain thalamic connectivity gradients and their neocortical projection maps. Second, genetic transcriptomic analysis was implemented to interpret the neuroimaging results. And finally, we use the generative network modeling and a novel perturbation analysis to these models to try to understand the underlying mechanisms of cortical network formation. In order to cover the wide developmental stages spanning from infancy, childhood, adolescence to young adulthood, we utilized two large open source datasets from the Human Connectome project. Below is the demographic information for the sample that we used. So going on to the results, during infancy, the first thalamic connectivity map, or CMAP, showed an anterior to posterior gradient, while the second one showed a superior to inferior gradient. And these are the neocortical projection maps, or neomaps, of the thalamic connectivity gradients. So you may wonder, what is a CMAP and neomap? The CMAP is derived from a dimensional reduction process to provide a summary measure of the high-dimensional functional connectivity patterns between the thalamus and the rest of the neocortex. Then, the CMAPs are spatially regressed against the thalamic time series, which are then multiplied with the cortical time series to project them onto the neocortex. Simply put, you can understand that regions that have similar colors have similar connectivity patterns. Now going back to the results, we observed that the first neomap showed a preliminary hierarchical network with the low-level sensory regions, the somatomotor and visual areas that were separated from the rest of the brain. Now, the second neomap did not seem to resemble any of the well-known cortical gradients, but interestingly, it showed high relevance to genes involved in developmental processes, such as neuron development, cell morphogenesis, and so on. Moving on to childhood and young adulthood, the neomaps exhibited more differentiated patterns compared to that of infancy. And in both maps, you can see that the salience network is consistently positioned at the higher end of the gradients. However, in the first neomap, the visual and somatosensory networks are situated at the lowest end of the gradient, and in the second neomap, the default mode network took that position. This suggests that the thalamus may play a role in differentiating between the internal and external axis of brain functioning. And these results were corroborated using a targeted gene approach where we tested if these neomaps can be attributed to the distinct patterns of underlying thalamic projections based on the relative density of core and matrix cells. Indeed, we found a significant association that reflects the external to internal axis with the salience network situated in the middle of the distribution. Now, finally, to gain a mechanistic understanding of how thalamocortical connectivity contributes to the formation of cortical networks, we explored a series of generative network models. These models create synthetic networks according to predefined wiring rules mimicking developmental processes. And then the simulated data is evaluated by examining the model fit with the empirical functional connectivity data. Next, we perturb the thalamus-salience connectivity in a series of growth models which incorporate age-dependent developmental information for network simulation. And we particularly targeted the thalamus-salience connectivities based on the previous neuroimaging results. For model fit evaluation, we used this metric where lower values indicate better fit. During infancy, gene expression and thalamocortical connectivity rules showed similar performance, while during childhood young adulthood, the thalamocortical connectivity-based rules showed the best fit to the empirical data. Next, using the simulated data, we extracted cortical gradients, calculated the segregation index, and assessed network modularity. I won't go into the details here, but these various measures substantiate the importance of thalamocortical connectivity in the emergence of large-scale organization and segregation between the external and internal processes. Finally, as I mentioned before, we applied perturbation to the thalamus-salience network in the wiring rules provided for the generative network models based on our neuroimaging results. And we found that perturbation to this connection compromised the segregation of both the salience external and salience internal axes. However, as you can see here, the salience internal axis was more dramatically affected, suggesting a higher contribution of the thalamocortical connectivity to the development of internal processing areas such as the default mode network during youth. Moreover, from these simulated cortical gradients, you can see that there is a more detrimental impact on the hierarchical network formation when applying the perturbation at a later stage of development, especially after 12 years. Now, going back to our research question, what is the role of thalamus in shaping brain functional architecture across development? Well, our study results highlight the dynamically shifting role of thalamus in shaping functional cortical organization across development. Specifically, to summarize, using these three methods, we found that during infancy, it contributes to establishing a preliminary hierarchical network while showing associations with cortical genes underlying development. Meanwhile, during childhood throughout young adulthood, thalamocortical connectivity plays an important role in functional network specialization, especially regarding the differentiation between the external and internal systems of the brain. Now, this brings me to the end of my talk, and I would like to give a special thanks to my mentors and collaborators who helped make this study possible. Thank you for your attention.